So this is to use its several few number is also called in the plants. That is called a important part of the organism. This which LAMPBRUSH chromosome amphibians are oocyte are immature eggs. Eggs. These are the one loop represent one chromated example DNA molecules area loops. It is a paired LAMPBRUSH chromosomes, but here a charismata, the region of the single chromosomes. So this part is also radicular part is also enlarged in the single part of the chromosome. This part is so which is also separate the small region of the chromosome showing a sister chromatid. This chromatid joining adjacent chromosome parts, which the chromomers are formed from the highly condensed chromatid. This is also axis. The similar to the plants or similar to the animals. This center part is also centromere like. Then either side is also chromatid or puff or external chromatin loops are there here. So this each one is similar to the chromatid or each one similar to the chromosome of a plants or animals, normal type one. The size of the loop may be ranged the average of a 9.5 millimicron to about 200 millimicrons. The pairs of loops are produced during the uncoiled and coiled parts are there. So this is also uncoiled and also coiled part is both are present. Present the highly coiled state in the chromosomes. Highly coiled state in the chromosome. So this is the complete figure of the LAMPBRUSH chromosomes, also called a polytene paired LAMPBRUSH chromosomes, which is also called a chiasmas, or the region of the single this part. But here, this single part, this radicular small single chromosome-like part is enlarged here. This size is also 10 millimicrons, one loop. Then either side of the two loops are between two loops are joined in part. The parting is called axis. This axis, which have consist of several number of the loops to either sides. That axis are spirally to each other of a other type of the loops of chromosomes. This is also clearly observed in the parts of the that the large have extended DNA looping, the formed through the DNA replication without separation of the cell division found in a oocytes of the parts. This one is also the LAMPBRUSH are seen in amphibians. This one same, but here is also complete this structure. So this one, these are the largest known chromosome, which is also character features are common in the found in amphibians or repetitions are there. Thus, they can seen a naked eye under the fine lateral loops arising from the chromosome during the first prophase. This is the first part. First prophase or diploid of meiosis. So during the meiosis, the prophase, which can divide into the 5 substages, that is also called leptotin, zygotin, PGT, diplotin, diagenesis. So in these substages, we are observed in the diplotin or leptotin regions are observed. So in the yak of the setting, which fish has amphibian with the loops have, it can be seen the naked eye characterize the fine lateral loops arising from the chromosome during the first prophase, also diplotin, leptotin, diplotin, diagenesis. Zygote diploid in a meiosis, special substages which can seen under the light microscopes, which are eyepieces, high level eyepieces. These loops give a brush-like appearance. That is why these are called the LAMPBRUSH chromosomes. Brush. So this brush means the chimneys are cleaning part. That is called a brush. The first discovered by these scientists are Fleming and Rookers. It consists of a longitudinal axis from the base single DNA molecule, which are several parts are there. This is also, it consists of a longitudinal axis of either side of the longitudinal axis, a coil structure that is called loops, a two loops, which is jointed part is called axis, the DNA molecules, along which several hundreds of bead-like chromosomers are distributed in linear fashions. So from each chromosomers, there are emerged to the symmetrical lateral loops, one for each chromatid, which are able to expand or contract in a response to the various environmental conditions. So about the DNA percentage of the each loop or each polyethylene chromosomes, 5 to 10% of the DNA it in the lateral loops. The loop formation reduce the mass of the corresponding chromosomes. So it is also amplifying the spinning out of chromosome material into later strands. The centromeres also have a appearance of a longest, they say, full and positive chromosomes, but so they are characterized by the lack of the later loops. This is also the LAMPBRUSH chromosome can be dissected into the from oocyte to nucleus. Individual chromosomes are liable to the strengthening individuals. So with extreme stretching, chromosome begins to separate transversely. Separated means each loop is also separated into the two halves, that the paired loop form a double standard bridges. The axis between the chromosomers is also doubled, which can be seen in the character features of the organisms, character features. Then it can certain special regions where two elements are separated longitudinally and here a single loop are there. This is also one character. What are the function of the LAMPBRUSH chromosomes? The LAMPBRUSH chromosomes, which can important for the synthesis of RNA. Then function of LAMPBRUSH chromosomes involve the synthesis of RNA and protein by their loops, and every loop to either side of the a single axis. So these loops consisting part is more percentage of the DNA which are located, that RNA is synthesized only at the insertion and then carried around the loops to be thick insertion. There it may be either destroyed or released into the nucleus. It is also first function of the synthesis of RNA by LAMPBRUSH. So the second one is also formation of the yolk material. So it is important of the, what is important of the yolk means there are some probabilities that LAMPBRUSH chromosomes are helping the formation of certain amount of yolk material for the egg to nutrients, which is also yolk present. That is also one. These are so you know that these are a polyethylene. The total bonds in a 5,000 bonds are size of each bond is also 3 to the 150. That is also important. But here found in a salivary glands, which are shows that. So this is also one bond structure of the polyethylene chromosomes are a cystodomia, serotonin, showing a nuclear part. Here is also nuclear part. These nuclear organisms, these are a heterochromatin part, also nucleus. But here interbonds and bonds. These bonds are black color parts, and either side of the bonds is called interbonds. These two parts are present in the, so but this is also, then it can normally polyethylene chromosomes in Drosophila. The structures are different. They're different. But here polyethylene chromosome from a Drosophila salivary glands are present in special type of organisms or salivary glands. These polyethylene chromosomes are which are several structures are there. This one, but it there from the formally many parallel parts during the larval development. The specific areas on polyethylene chromosome become uncoiled and also puffs are present. These special parts are there. So these puffs and ball beanie rings. It is a special. During their initial stages of development, these bonds on interbonds of the chromosome exhibit a swelling or puffs. Swelling means that the size of the bonds is also increases, which is also called a swell. That part is got a puff. Their appearance it depends on the stages of a larval development. So it is a probability that the metabolic activity is required for the formation of the puff. Therefore, the puff is also special parts which can be increases in the size and also performing the secretory function of the salivary glands. The formation of this control by the certain specific genes and the puffs are related with the active synthesis of RNA and protein. But the functions are, that these two type of the functions are also three functions are this chromosome also help the proteins in these heterochromatin regions. Then also called shifting heterochromatin and in respect to the euchromatin produces gained. The main function of the polytenic chromosome to carry genes, which ultimately control physiology of organisms. These genes are a form of the DNA molecules, but that is also important of the chromosome puffs. It is also gained chromosomes, but here is also endocytosis. Then LAMPBRUSH chromosomes are, which is also major structures there. This is also LAMPBRUSH. Then also LAMPBRUSH also polytene chromosomes. Then B chromosomes. The B chromosomes are a special type of the chromosomes which can located in the organisms. Then the organisms. Here is also the polytene chromosomes bonds are there, but here LAMPBRUSH chromosomes are completed, but each loop is also complete. But this is here is also B chromosomes or a supernumerary chromosomes. That it is a accessory aberrant of redact chromosomes, extra copies of normal chromosomes, extra copies. So not homologous to normal chromosomes, but and are not essential. So normally they are highly heterochromatic regions. Highly hetero means also different characters. Then more the DNA percentage based to euchromatin and heterochromatin two parts are separated here. So they are present in the many species of animals and from flowering plants like a maize, rye, some ants, some grass, hoppers, etc., which are special parts that is got a B chromosomes. So these they are increased presence in any individual organisms produced the marked phenotypic effects that so they are often lost of the gained in any individual organism. Some features are small in size. The special features of a B chromosome is called a smaller in size, heterochromatic, allocyclic, and so telocentric or acrocentric. It is special part. Then it can expedite structural polymorphism among cells and tissues of same organisms. Then the genetic effects is less compared to other chromosomes. Less compared to other, it can present in a large amount of the effect, fatality and vigor. It is a one character. Then relatively unstable. It can get eliminated due to the latching and non-disjunctions. So this non means also Mendelian inertancy, no chiasm appearing. These the special features of the character features are present in the B chromosomes. That is also the structure of the B chromosomes are different here. So this one, the chromosome 4 attached to the spindle. attached to the spleen. Then it is also breakage in a centromere regions. Breakage of centromere. But here centromeres are mid divisions. Then fusion of the broken short arms to create DNA isochromosomes. This is also special parts are the structure of the B chromosomes are here. Then you A, but significance of the B chromosomes it affects seed and pollen fertility. It is present in the brains. It affects the seed and pollen. Also, flowering time is delayed. The flowering, non-flowering and flowering times are delayed. Vegetative growth of plant is also get adversely affected due to the presence of these chromosomes. It also affects the pairing of a chromosomes, resulting in special hybrid process. Special type of the hybrid in suppose in suppress or a promote meiotic pairing in integrating the interspecific hybrids. This is also important of the character features of the B chromosomes. That the B chromosomes are the special quality in also called. But some of the regions quality in also LAMPBRUSH chromosome. It is also then structure of the polyethylene. Then LAMPBRUSH. Here is a LAMPBRUSH chromosome separated. This is also LAMPBRUSH chromosome. It is the longest chromosome in all. It can present in the nuclei of the loose animals. Loose animals. But the significance of the B chromosomes here is also special chromosomes in the brains. Dash chromosomes are a unusually large chromosome found in a certain insect salivary glands, that is polyethylene. The character features of binding pattern of polyethylene chromosome is due to the repeated round of dash DNA replication is there. DNA replication. The LAMPBRUSH chromosomes are found in the dash oocyte during the meiotic process and also got amphibians. Then polyethylene chromosomes are most famously studied in the genus dash Drosophila. Then the lateral loop in the LAMPBRUSH chromosomes are see at the sides of active means transcriptions. So then the large loop in amphibians or LAMPBRUSH chromosomes are sites in the transcription dash. The chromosomes are formed when chromosomes tail to the repeated during the cell division. Also B chromosomes are superannuary chromosomes. In polyethylene chromosomes, the dark bands repeated regions of dash means also condensed part. The visible puffs on the polyethylene chromosomes are called dash chromosomes puffs. So the chromosomes puff. Then the LAMPBRUSH chromosomes were first observed in the scientist is also called with Fleming. Ether Fleming. The LAMPBRUSH chromosomes are typically found in the dash. It's also diploid. These chromosomes were first observed by the Walter Fleming. LAMPBRUSH chromosomes are named due to their resemblance to a dash. It's a lamp brush. The lateral loop of the LAMPBRUSH chromosomes are sites of active. It's also transcription site. The LAMPBRUSH chromosomes are most commonly studied in dash. Means also called the amphibians and birds. Amphibians and birds. The extended lateral loops are rich in dash. It's also messenger RNA, mRNA. Each bivalent in LAMPBRUSH chromosomes consist of dash. Four chromatids are there. Four chromatids. The LAMPBRUSH chromosomes are absent in most dash means somatids